I think we're gonna wait just another moment. I think Walter will join us, and
I hope so, yeah.
so, yeah, thank you guys for joining, and thank you all for joining in person too. It's really exciting. And Tiffany, can you give me the ability to share my screen?
I should have already. I just clicked it, so. Oh, is that Sam that's coming in too? Hi, Sam.
Hey. Gang's all here, oh my gosh.
What a beautiful title slide.
Good, you can see it. Yeah, okay. Nice, great.
We're just waiting on Walter then, yes?
I think so, yeah.
Okay.
I think we'll wait like 3 minutes and then just get started. If not, we'll... Do you want this time? Do you want it?
And Amy, Beth, and Martin are here. Hi, guys.
Okay. So, sorry, it's gonna be stuffy. Just get started.
He's 2 minutes late. Andy's German, so I'm not sure what's going on.
How is everyone doing today?
Aspen, I'll go ahead and send Walter a reminder email.
He's actually on time. His clock's right. Yeah. Wouldn't it be embarrassing if I forgot to send him the link?
I think that's impossible. So I did send him an email. And I think we should start in 1 minute. If he doesn't.
Okay.
Give him 1 more minute.
I do a bag of candy if I want this. I really don't like to make food.
Aspen, why don't you go ahead and get started? I think he's not coming, or he'll be quite late. So.
Yeah. Make sure this works. Great. So again, thank you all for coming today. I'm really excited to share with you guys the topic of my project study, which will then be the topic of my master's study. That is population genetics of Lupinus tedesstromii and Lupinus camastonis across both species' geographic ranges, and this work was done under the supervision of Tiffany Knight and Walter Durwell. That's not working. Great. To start out, we know that species are at risk in the Anthropocene. Current estimates say that approximately 25% of species are at risk of extinction due to human activity, and current extinction rates are tens to hundreds of times higher than background rates and are only expected to increase unless we do something. Major threats to biodiversity include the introduction of invasive alien species, overexploitation of habitats, pollution, anthropogenic climate change, and habitat change, which we can further break down into habitat loss, habitat fragmentation, and habitat degradation. Now, we normally think about these drivers acting on the scale of species diversity, but within the context of an individual species, these same threats can also threaten the genetic diversity of a species. Now, things like habitat fragmentation, loss, and degradation can lead to bottleneck events, increased genetic drift, increased inbreeding, and decreased gene flow between populations, which can lead to the loss of genetic diversity and increase the extinction risk. The loss of genetic diversity increases the extinction risk in several ways. First, the loss of heterozygosity reduces individual fitness, which has implications for the total population viability. Additionally, selection can only act on the variation that exists within a population, and when you lose the genetic diversity, it's less likely that individuals in the population will respond differentially to changes in environmental pressure, and therefore the species' ability to respond to changing selection pressures is also reduced. This can all contribute to the local extinction of populations and therefore also contribute to the total extinction of the species. Populations are increasingly at risk for extinction due to the interdependency between population size, genetic variation, and fitness. So consider a loss in a population. You're also losing not only the individuals, but also the genes that they represent in the population, which can have implications for the fitness of the population and further drive losses. We refer to this interdependency, ever-decreasing interdependency between population size, genetic variation, and fitness as the extinction vortex. External threats such as environmental stress from anthropogenic climate change can exacerbate the negative effects of decreased genetic variation and cause species to spiral more quickly. Additionally, species caught in the extinction vortex are at an increased risk of things like outbreeding depression and genetic swamping, which are negative processes of hybridization. The species that are most at risk are characterized by being those that are already rare, those with inbred populations, those with really small populations, and those populations are isolated from one another. Now, one such species is Lupinus tedesstromii, a small plant endemic to coastal California. This plant was listed as endangered in the 90s as its habitat was being destroyed for the construction of golf courses. Currently, the species occurs at approximately 10 population centers at 3 locations across coastal California, with around 1 million individuals total. This is thanks largely due to extensive restoration efforts that have happened at Point Reyes National Seashore, which is the center point there. And so there's about the majority of those 1 million individuals are occurring there. But as you can see, populations are spread pretty far apart, and we suspect that there's very little to no gene flow occurring between them right now. This species has also been part of a long-term monitoring project since 2005. It's a very unique data set, a very valuable demographic data set. It's very special for an endangered plant like this. And from this work, my colleagues and advisors have found that it is essential that Lupinus tedesstromii has early successful microhabitat in order to persist long-term. These same microhabitats are threatened by the introduction of invasive species such as European beachgrass and ice plant. But studying the species for this long has allowed my colleagues and advisors to find out that it's not just the fact that the beachgrass is coming in and taking up space, but it's actually also providing habitat for the native seed predator, mouse, which is consuming the seeds at a much higher rate in invaded areas than in early successional habitats. Here we can see little mouse's cache of seeds that it has been snacking on right outside of its burrow. This long-term data set has also allowed my colleagues and advisors to assess the efficacy of restoration to remove the European beachgrass, and they have shown that indeed removing the beachgrass decreases seed predation. But in addition to the direct and indirect effects of invasive plants as a risk, Lupinus tedesstromii is also at risk from changing climate. This graph here shows the population growth rates of 7 subpopulations at Point Reyes National Seashore, We can see that if the average annual temperature increases from 12 degrees Celsius to 13, all the population growth rates are expected to drop below 1, the replacement rate. And in fact, this population represented by the lowest line here in green has already decreased to 2 individuals since the study was published 2 years ago and may pretty much be effectively extinct already. So we know that this species is under a lot of external stress. It is essential that we act quickly to make management decisions before we lose it altogether. Now, however, a lot of management has already been happening at Point Reyes National Seashore, but it has had mixed results. So some of the populations are doing well. They've exploded, there's hundreds of thousands of individuals, they're continuing to increase following restoration. But some of the populations are doing not so well. Some of them are remaining very small or even continuing to decline following restoration. And it's thought that perhaps these populations are already experiencing critically low genetic diversity and processes such as inbreeding depression that might be driving this poor response. Land managers are, however, considering the translocation of seeds from the northern subpopulations to the southern subpopulations in order to supplement that gene pool. But it is important that we understand the population genetic structure of the species of the entire range to assess whether or not this type of action would actually be beneficial to the species in the long term. However, one other thing that complicates the situation is another species of Lupinus camisonis, which naturally co-occurs with Lupinus tidusstromii at the northern subpopulations at Point Reyes National Seashore. However, Lupinus camisonis was also introduced to the populations that occur at the Monterey Peninsula. And this hybridization could pose a threat for our seed translocation, as the Lupinus camisonis does not naturally co-occur at these southern subpopulations at Point Reyes. But by studying the populations at Monterey, where it was introduced, we might better understand the risk of accidentally introducing some hybrid seed in case we did that in the seed translocation. So in summary, we know that Lupinus tidusstromii is rare. It has really small populations that are pretty isolated from one another, and it's under a lot of environmental stress. However, what we don't know at this time is whether or not these populations are inbred, whether or not hybridization poses a threat, and if any of the populations are already suffering from critically low genetic diversity. We would like to know if genetic processes such as inbreeding might explain why some of these subpopulations are responding so poorly to restoration, if hybridization with Lupinus camisonis threatens the long-term persistence of Lupinus tidusstromii, and which populations of Lupinus tidusstromii harbor the most and least genetic diversity. Now, we ask all of these questions within the context of a real-world management application. Should we translocate seeds from the northern subpopulations at Point Reyes to the southern subpopulations? And in order to explore this question, we're using a within and among population genetic structure framework to better understand the genetic diversity of the species across its entire geographic range. We will examine the hierarchical structure of the species and also use isolation by distance analysis to explore the relative contributions of genetic drift gene flow to the genetic structure across the whole range. Then we will look at the within population genetic variation to understand how these populations are structured at subpopulation level, looking at the expected heterozygosity and the inbreeding coefficient of individuals. Finally, we will apply the same framework to Lupinus camisonis to better understand how that species is structured across its entire geographic range, detect cryptic hybrids, and assess the relative risk of hybridization across local species ranges. With this framework, we will be able to make recommendations for seed transfer and restoration actions to best benefit Lupinus tidusstromii at Point Reyes National Seashore. For our data collection, I recruited a team of volunteers to help me sample from all of the sites on Long Coastal California. We were able to sample from sites where only Lupinus tidusstromii occurs, sites where only Lupinus camisonis occurs, and sites where both species co-occur and are known to hybridize, including sites where they co-occur naturally, sites where they co-occur because Lupinus camisonis was introduced. At each site, we sampled 12 individuals along a transect, and from each individual, we sampled a handful of leaves and took plenty of photos. Next, we will extract DNA from the leaves, generate dd-RAD libraries, send the samples off for sequencing, we get the data back. We'll identify and genotype single nucleotide polymorphisms, SNPs, using the DOSIM pipeline. So the results of this work will allow us to make a concrete recommendation for the management of this tidusstromii at Point Reyes National Seashore, but it will also fill an essential gap in the knowledge of our extensive knowledge of Lupinus tidusstromii and allow us to make decisions about the management well into the future. Additionally, this will contribute to the field of conservation genetics by serving as a unique example of within and among population genetic structure of an endangered plant threatened by human activity, And finally, this work will serve as a stepping stone for future work where we can combine it with those population models to better understand the variation between those subpopulations and with the climate models to understand how the species will respond to climate change. Lastly, it's important that we continue monitoring these sites of hybridization to better understand this risk. This work will serve as a first risk assessment of hybridization and allow us to start detecting those hybrids in the field much better. With that, I would like to say thank you guys for listening today. Thank you to Tiffany and Walter for supervision and guidance, and I'm happy to take your questions. Wow.
Questions for Aspen, please. I can't see who's there in person. I don't know if Aspen, you want to moderate the questions and can keep track of the in-person and the online? Because there's more people in person than there are online, I think.
Yeah, there's a lot of people online too, though.
Okay.
No, I don't. So they're definitely fertile, we know that for sure, but we don't really have an understanding of how their reproductive rate compares to either species at this time. So that is something that we'd be interested in looking into in the future for sure.
Aspen, if it's not too much trouble, can you repeat each question before you answer it, just so everybody can hear it?
Yes, to make sure I'm asking if the hybrids are fertile and how they compare, how the reproductive rate compares to what species. Yes. Sorry.
Thanks. Can I ask a question?
Please.
So in terms of, I think it was a really nice presentation and a really nice broad introduction to the field. What I wanted to know is a bit more about the novelty of the research you're doing for your master's and how it fits into this field of study. What would you say is the most important contribution in terms of the newest thing or the most exciting thing in terms of the novelty for the field?
Thank you for your question. I'm thinking, in my personal opinion, I'm most excited about the hybridization aspect because there's not a lot of studies that look at hybridization as an extinction threat for plant species. However, that is not the focus of this work. And I think the most exciting part about this work is the fact that it is going to directly inform management of the species and have implications that we can then track with the population monitoring well into the future.
I think so too. I think there's often studies that look at population genetic structure, but I think few where they're asking applying it to a direct management question. So I think that's also a novel part of this.
Thank you.
When you were talking about relocating the seeds and the possible threat hybridization would have, I was wondering, is there any knowledge about, for example, local adaptations of the native one or the congener, the endangered one and the congener, so that there's also might be the threat that if it comes to hybridization, that the populations would exceed the endangered one?
Yeah, so Arhan is asking about levels of local adaptation in case we were to accidentally introduce hybrids from the north to the south of Point Reyes. And I think that's something that we are definitely interested in. It's not something that we know at this time, but I think this work might be able to tell us at least a little bit about how unique these populations are from a genetic perspective, and combined with perhaps the responses to the restoration. I think there's perhaps some evidence that there is maybe some local adaptation, but also the scale in which this translocation would occur is, and it's pretty small in the grand scheme of things. I mean, it's a handful of kilometers. It's not like the climate is actually that different, or even the microclimate is that different. Does that answer your question?
Yeah, totally. I was wondering if the southern populations would have a different microclimate than the others.
I would think not, yeah.
So you mentioned that you don't expect that much gene flow between between populations and what they should be able to be able to spread.
Yeah, so Sana's asking about gene flow between populations and whether or not we know much about the dispersed. Gene flow, like cross-pollination between the subpopulations, but they've got pretty heavy seeds. So these seeds, when the pods ripen, they pop and they explode, and the seeds go flying like within a meter or so. But then they're turned over by the sand, and they're maybe going to be dispersing a couple of meters, but I don't think the seeds themselves are going to be traveling long enough distances to introduce new individuals to other populations. Additionally, they are pollinated by bumblebees, and it's a very windy area. And so even bumblebees that are really good, really effective pollinators and really good flyers too, there's kind of a limited ability of them to travel long distances along the coastline. So I would expect that they would not really be able to bring, certainly pollen hundreds of kilometers, but even within Point Reyes, I think it might be a bit challenging for bumblebees to travel between some of the populations that are spaced further apart. So that's your question.
That also sort of brings up the next question. Is it, if there's no gene flow between the different populations, and you do introduce seeds to increase genetic diversity in the populations, assuming it's low at this point, isn't it sort of going to be a never-ending quest of like, you have to keep on introducing new genes, or do you expect there to be enough individuals in a population to retain that genetic diversity? So I think one of the long-term goals might actually be increasing connectivity between these patches, so that gene flow can occur between them. Because I mean, if we just bring in some and then 50 years later the genetic diversity is back down to where it is now, how helpful is that? But also, this is a species that we're really running out of time with these climate projections. We know that we're already losing these populations, and doing something seems to be better than doing nothing.
That was my question. Might it be fair? What are the predictions? Do we have that down?
Yeah, I mean, if we look at strictly the climate predictions, can I do that again?
No, my sense.
Sorry, I'm sorry. All my little animations, they're very annoying and backwards. Okay, there it is. Okay. Yeah, so this was a little paper, and we know that the population growth rates, if you get one degree increase in temperature, which is projected to happen, I think by 2050.
21?
2050, sorry. Yeah.
Well, whatever. It's coming up fast.
Yeah. Like these populations, they're going to start collapsing due to climate effects only, and that's not considering any other like genetic effects, anything else.
Vera.
And
Vera.
like I said, we already have almost lost this one population. It's 2 individuals, and that's not functional.
Did you say 2? Sorry.
Yeah. So it's
Is there a seed bank?
is there a seed bank? Really, really a good question. So the seed bank dynamics of this species have been studied, yes. And there is a really good seed bank, and we suspect that, like in the short term population dynamics, that it's about a 2-year seed bank. Seeds aren't going to germinate immediately, although they could germinate that same year. It's often that they're going to remain in the seed bank for 1 or 2 years. However, the sand in these ecosystems is incredibly dynamic, and it's going to be moving and covering up seeds, burying them so deep that they definitely won't germinate. And another big wave might come in and bring those seeds back up to the surface and could serve as maybe a rescue. But with the restoration at some of the more northern populations at Point Reyes, they found that just by clearing away the sand, this plant was sprouting. Seeds are there, for sure.
Just that the seedlings are never done.
Seedlings are going to die. They're going to be broken. there used to be more connectivity. There used to be other populations of Leucanthus stromiae that occurred between Monterey and actually further north of our most northern population, I believe. But a lot of this area is perfect beachside development. It's easy to build a house or a golf course on, and so it was targeted for human development. And so that's why a lot of them were lost. However, the Leucanthus camsonis has a much broader range. What is that? Sorry. Yeah, so the blue dots on this map are the range of Leucanthus camsonis, and so it's occurring in a wider ecological niche. That due to plate tectonics, the Point Reyes Peninsula has been sliding north along the coast, and so it has very different ecological properties than the mainland right across the bay from it. And it's also very similar to Monterey Peninsula. So there used to be other populations with similar areas along the coast, and we've lost them. Yeah, yeah. So one of nature's asking how these species are kept separate. And I think part of that is phenology. So they overlap in flowering quite a bit, but their peak flowering times might be slightly different. But also it's mostly their habitat. So the Leucanthus stromiae is definitely an early successional species. It is the first thing to come up when sand is blown out. Whereas the Leucanthus camsonis it's shrubbier, it's much more a later succession and also more of a back dunes species. So they're not really occurring in the same exact area either, even though they have like the same the range overlap. They might not be occurring at the exact same scale. Do you have any questions online?
This isn't a question, it's a comment. A lot of us saw your presentation last week, which was very good. Every question I had in my mind, which was dependent on either my knowledge or all the things that you were doing, you pulled it all together, and I feel like this could be dangerous, but I feel like I understand. And you did a really amazing job. It's really, I can't believe you clearly heard what people said, and you clearly understood what you needed to do. It's just amazing.
Thank you guys for the feedback. To that, it was really, really helpful. And it means a lot. Obviously, helps.
While you were talking.
It's not.
Are we done with questions? Nobody has any other burning questions? Okay, then I believe we can move on to the next phase, which,
Yeah.
There's one question.
One quick.
oh good, one more question, go.
How is this distinguished from the species in the field?
Okay. Karina's asking how we distinguish the species in the field, and it is a lot of it's by field. No, there's a lot of morphological traits that are different between them, but with these hybrids, it's actually really, really hard to tell because, obviously plants are very phenotypically plastic anyway, but when I was there in June, one of the biggest indicators for me was actually phenology. Most of the Leucanthus stromiae was done flowering, it was done fruiting, whereas the Leucanthus camsonis was in full flower and full fruit. But the Leucanthus stromiae is much more prostrate, it's spreading on the ground, it's pretty small. Leucanthus camsonis is a bit more shrubby. The flowers of Leucanthus stromiae have a little bit of yellow on the banner, Leucanthus camsonis does not. The leaves are a little bit different too. It's easy, especially when the individuals are big, but when they're small, it can be kind of hard to.
Yeah, thank you.
Great. Now we can move on maybe to the next phase. And I believe that is, I don't have as much experience with this. This is the grading phase. And I believe you're not supposed to be present, Aspen. But I need your computer to be present so that I can be present. And then I believe anybody that has a PhD can participate in the grading. Is that correct?
I think so, yeah.
Okay, and I am not sure if there is anyone online from MLU. Sabrina Traeger was here, but now she's not. And so it might just be me online. And then I need to be able to communicate with all the people in the room. And I believe there's also a form, Aspen.
Yeah.
That there's a form. Did you can email it. Okay, good. Do you have a printout with you there?
No, but we can arrange that.
That's fine. No, that's fine, Aspen. I think that's fine. And so maybe you could arrange it. So, well, I guess since people are now leaving the meeting that are online, we should thank you all for coming and participating and for your questions and for your support of Aspen, because I think you're all going to leave. And then, yeah, Aspen, you can move towards making sure I can communicate with the other. I think we needed at least, there needs to be 5 people grading, at least 5 PhDs. I hope they're in the room. Me and 4 more. Yes?